Hi everyone. Let's just wait for maybe 2, 3 more minutes for a few more people to join, and then we'll get started. Hmm. Okay, I will share my screen. Just let me know when you guys can see it, okay? Mhm. Can you see the screen?
Yes.
Okay, great. Cool. So I think we can get started on today's session. So this was a very interesting week of coursework, I think. It was meiosis as well as a lecture on organization. And yeah, I have some theory parts as well as a whole bunch of question answers that we can go through together, which would hopefully help you with your assignments and later on if you have registered, then for the exam as well. Let me know if you have any questions in between, and you can stop me at any time. Okay. And I think until required, try to keep yourself muted. That would just help to make sure there's no interference there. And of course, when I read out the questions, if anyone doesn't know, what we do here is we typically, I'll read out the question and you guys will say what the answer is. So there'll be multiple choice or fill in the blanks or whatever, and then you can say the answer. Okay. All right. So let's get started. So the main part of your coursework for week 5 was meiosis. So what is it and why do we need it, right? Because we've already studied about mitosis in so much detail. We looked at, how cells divide and form identical daughter cells. So why do we need a whole other program of dividing cells, right? So meiosis is a type of cell division in sexually reproducing organisms. And what it does is two main things. One is that it reduces the number of chromosomes in gametes. So these gametes are basically the sex cells like the egg or the sperm, which will later fuse during fertilization. And this reduction in the number of chromosomes in the gametes is required to maintain the correct ploidy level. So as you know, in fertilization, a sperm fuses with an egg, right? So if you have diploid cells fusing, that would mean you have way too many chromosomes. You'll have 4n in the cell, and that's not right, right? If we are diploid organisms, that would mean that the gametes would then need to reduce to n. So that's one thing that needs to happen. And the second thing, of course, is that during the process of meiosis, particularly in the phase meiosis 1, there is some genetic variation that is introduced in the form of homologous recombination events that take place between non-sister chromatids of a homologous pair. And I'll go into detail of this a little bit more in the upcoming slides. So meiosis is needed to maintain the correct ploidy and also create genetic variation. And in humans, in the body, somatic cells are diploid, as I mentioned. So they have two sets of chromosomes. So to maintain this state, the egg and sperm that unite during fertilization needs to be haploid with a single set of chromosomes. So during meiosis, each diploid cell undergoes two rounds of division, so meiosis 1 and meiosis 2, to yield four haploid daughter cells, which are going to be your gametes. Okay. So just sort of reminding or essentially just going through this so you guys kind of brush up and remember whatever you have studied till now. You'll have one diploid cell here, and then its chromatin has duplicated in the S phase, just like how we have seen earlier during interphase. And then it enters into meiosis 1, which is the first phase of overall meiosis. Within meiosis 1 in prophase 1, and we'll go into the sort of subdivisions of prophase 1 in the upcoming slides as well. You have your homologous chromosomes that start condensing and they start aligning together. And then in metaphase 1, they join up at the metaphase plate. However, here one interesting thing that's different from mitosis happens, which is that here we are not separating the chromatids from one another. So the chromatids, the sister chromatids, essentially whatever has been replicated during S phase, the same two chromatids are going to go in the same side of the cell, right? So your paternal chromosomes and your maternal chromosomes are going to be separated. Of course, they have recombined, so there is some mixing of genetic material. So it's not 100% paternal on one side and 100% maternal on the other side, right? So just these homologous chromosomes with the sister chromatids attached together at the centromere that is intact in this phase, these homologous chromosomes are separated are attached at the microtubules in metaphase 1. And then they come apart and they're separated in anaphase 1. And then, of course, you have whatever telophase 1 and then cytokinesis, and that leads to your two haploid cells. So already the reduction has occurred and you already have half the number of chromosomes. So the next phase, meiosis 2, essentially ensures that you are ending up with four haploid daughter cells. And this is when it undergoes prophase 2, where now you finally have essentially again you have this decondensation, and then this condensation in prophase 2, and then you have the separation of the sister chromatids this time around, and that occurs in anaphase 2, and then telophase, and then you have 4 haploid cells, each with the single ploidy of each chromosome. Okay, so I'll go into a little more detail. Of course, the coursework was great, so I'm sure you guys have already understood all of this, but it's great to sort of jump through the concepts a little in case there was something you were confused with. This might help you. So, haploid, so what is this ploidy business, right? What is haploid? What is diploid? I'm sure you guys are already well aware. So, haploid means that there is one copy of each chromosome. So, in this case, the pink, blue, and green ones you can see, these are 3 non-homologous chromosomes. You can see their different sizes, right? Their, constrictions are in different regions. These are 3 non-homologous chromosomes. Diploid condition, or 2n, which is what most of our somatic cells are, this means you have 2 copies of each chromosome. So one of them, so in this case the purple and the pink, one of them is of maternal origin and one is of paternal origin. And so these, why are they called homologous? That means that essentially the genes are the same and the positioning of the genes are the same across these 2 chromosomes. However, the allele of that gene might be different based on whatever is the phenotype of that individual. You can trace back and see that, okay, so maybe this one, there's a one dominant allele here and there's a recessive allele here of the same gene. But if you were to look at the positioning and the frequencies of these genes, it's going to be exactly the same. So it's the corresponding chromosomes, essentially, one from the mom and one from the dad. And that is the same case with these other chromosomes as well. So these are homologous chromosomes. And in this case, this is when it has not been replicated in S phase, right? So this is a single chromatid with its short arm and long arm. And this represents one chromosome, and this is the homologous chromosome. When you have replicated this during S phase, because we're going to divide it up into cells, right, evenly, that time you're going to have another purple and another pink, essentially, right, that are joined at the centromere. And that's just 2 sister chromatids, each of one pair of chromosomes, and those are homologous chromosomes of each other. So I hope that is clear, right? So the text basically says the same thing, that nuclei possessing pairs of homologous chromosomes are diploid, which is this 2n, and these nuclei will possess 2 gene copies, which are alleles, which may be same or different for each trait. And in the haploid condition, you have nuclei possessing only one set of chromosomes, and in that makes it haploid. And these nuclei will possess a single gene copy or allele for each trait. And all the sex cells, gametes in an organism, will be haploid, and they're derived from diploid cells initially via meiosis. So the gametes themselves don't divide. Once they're haploid, either your egg cells or the sperm cells, they don't actually divide. Once they're haploid, that's it. They come from these spermatogonia and these sort of germ cells that are present in the testes and in the ovary that allow for this kind of meiosis to occur, right? So that's haploid and diploid. So that is the basis of understanding how these chromosomes are segregating and what is a homologous pair and what is a sister chromatid or a non-sister chromatid. Okay. And this is just to sort of clear that up a little more, because I remember when I initially used to study this, it used to confuse me a bit. So I thought maybe if there's anyone there who's confused about it, this might clear it up for you. Right. So, in sexually reproducing organisms, as I mentioned, we inherit the genetic sequences from both the parents, right? So you have oops, my bad. Okay, as I mentioned, you have a maternal copy and a paternal copy, and these for instance, chromosome 1, you will have one from mom and one from dad, and they form a homologous pair. Same for the next set of chromosomes and the next and the next. So homologous chromosomes, they share the same structural features as I mentioned. So they have the same size, as you can see, the same band pattern. So like where the genes are, where there are introns, exons, all of that is the same. And the same centromere positionings. As you can see, the centromeres are in the same region. Um, and the same genes also are there at the same loci. But of course, while the gene is the same, so it's like if it's coding for hair, over here, both of these genes are coding for hair. But the allele for what kind of hair, what is the ultimate phenotype? Is it brown hair, blue hair? Oh, not blue hair, maybe black hair. Uh, that allele may be different. Okay. So that's the case with homologous chromosomes and sort of what they code for. And in here you can see, to clarify, that you have these homologous chromosomes with their centromeres. When they're replicated in S phase, each of these have a replicate copy. This is exactly identical. So these sister chromatids don't differ from one another at all. Okay, so these are sister chromatids of the same chromosome, and these are sister chromatids of another chromosome, and these and this are homologous chromosomes. Okay, so this is very important to understand, because in meiosis, in that process, we are separating both homologous chromosomes from one another, which is done in meiosis 1, and then we're separating the sister chromatids of a single pair also, which is in meiosis 2. Okay. So I mentioned the phases of meiosis earlier. So I'll go into detail now, particularly in meiosis 1. Um, Starting with meiosis 1, where the first major phase of meiosis 1, after we have moved ahead from G2 into prophase, is prophase 1. Okay. So in prophase 1, there's actually so much going on that we've divided it up into 5 different sub phases, which is leptotene, zygotene, pachytene, diplotene, and dikinesis. So in leptotene, and I have some nice images for you guys here as well, but I'll just explain the phases in brief before we go ahead to that. So in leptotene, this is when your prophase is beginning. So just as in mitosis, you remember that in prophase, what starts happening, right? The nuclear envelope will slowly start breaking down, uh, and chromosomes start condensing so that they're actually visible now, right? So they're not thread-like anymore. So chromosomes start condensing, the nuclear envelope starts to break down, and now it's time to start aligning these chromosomes together. And why is that important? That you'll see in zygotene. So in zygotene is when this synaptonemal complex starts getting loaded onto these chromosomes, onto the homologous chromosomes. So, and that helps with beginning the process of synapsis. So what is synapsis? It's essentially the aligning of regions of homologous chromosomes with one another. And this SMC, or the synaptonemal complex, this is very important for essentially acting like a glue between the homologous chromosomes during this process. So that starts in zygotene. Okay. So loading of this complex happens in zygotene, and by the time pachytene is reached, uh, we have maturation of the synaptonemal complex. So all of the components have been correctly placed, all of the parts have been aligned, and the chromosomes are ready for crossing over. So crossing over starts, which is essentially DNA exchange that takes place between non-sister chromatids. So that's how we're going to have genetic recombination, right? If we have crossing over between sister chromatids, you're just mixing the same genes with the same genes. So nothing is going to happen. You need to exchange the genetic material between the maternal chromosome and the paternal chromosome. So those are non-sister chromatids of a homologous pair of chromosomes. I hope this is clear to everyone, right? If anyone is confused by these terminologies and stuff, please let me know. Okay, so in pachytene, we have this DNA exchange that's taking place, crossing over in these regions called chiasma. Okay, and in diplotene, synapsis ends and this chiasma is visible within this structure. Oh yeah, and this structure is called the bivalent, which is basically the linked homologous chromosomes. And then when we reach diakinesis, the nuclear envelope now is completely breaking down. So you have to remember that this is still prophase. So slowly, slowly through prophase, while all of these steps are taking place, the nuclear envelope begins to break down. So by diakinesis, the nuclear membrane disintegrates and prophase ends with the crossing over having taken place successfully, and then the cell will move on to metaphase. So I hope that is more or less clear to everyone. So now moving on to some nice images that I found in this paper, that this link is there below. This is in a plant cell, I think in a wheat plant. So they took cells from the anther of the plant. So basically you have these male sex cells that they have isolated, and they have done basic light microscopy to visualize these different phases of meiosis. And so let's go from A onwards. So you can see at A here, you can't really see the nuclear material very clearly. The cell is quite um, indistinguishable when it comes to any phase. Okay. And this is essentially during interphase. Is my mouse visible? Yeah. So this is during interphase. This is a pre-meiotic G2 nuclei. That's what they have labeled it as. And in B, this is leptotene. So as I mentioned, this is just the start of prophase. So your nuclear membrane is still quite intact. The chromosomes have not escaped into the cytoplasm just yet. They're just going to start sort of moving close to each other and start getting aligned in leptotene. Then C, this is zygotine. So in zygotine, you have the synaptonemal complex that starts aligning the chromosomes. So it's not the clearest image in the world, but you can see that these structures are sort of coming closer together to one another. So I guess if there were some more high resolution images or something that had more fluorescent markers with better contrast, maybe we'd be able to see um, this joining better or this alignment better. And in D, this is pachytene. So here essentially the complex has matured and all of the chromosomes have aligned with each other very well, and genetic recombination has started to take place. Then in E, we have diplotene, where the nuclear membrane you can see now has completely gone, and these chromosomes have sort of I don't know why they're so dark here, to be honest. I'm not sure. But essentially, they've completely condensed and they've completely aligned with one another. And they've formed these bivalent structures. And in F, this is diakinesis. Um, this is where it's diakinesis is going to early metaphase, so where they've started slowly moving towards the metaphase plate, which is more evident in G, which is early metaphase, metaphase 1. Of course, this is all part of your meiosis 1. Okay. So this is early metaphase 1, where they've started aligning at the metaphase plate. Here they've completely aligned in H. And then in I, anaphase 1, you can see that they've separated nicely. So this is the separation of homologous chromosomes, okay, not sister chromatids. So homologous chromosomes have gone to separate ends of the cell. And then in J, this is telophase. So they have separated fully, and we're going to start seeing an invagination of this wall over here. And in K, you can see that these are two separate cells now. And this is presumably part of the next part of meiosis, so meiosis 2. So this is when these two daughter cells that were the product of meiosis 1, they are now undergoing metaphase 2. So now we know that meiosis 2 is very much like mitosis, where you're separating the sister chromatids. The only difference is that the final product yields a haploid cell. So here you can see that they've labeled it as metaphase 2. So in metaphase 2, you have these chromosomes now aligning at the metaphase plate, and the sister chromatids will be attached to the microtubules and prepare for separating into daughter cells again. So this is telophase 2 here, where you can clearly see that there are 4 daughter cells, which all started from here, where there was just one single cell. So I hope the process of meiosis from both your coursework and whatever I've explained now is clear to you. Uh, and this is a video again from Drewberry Wahi TV channel. It's a really nice resource for visualizing these kind of things. It's a short video, and I hope you guys find it educational. So let's just go through it together.
Meiosis is a special type of cell division necessary for sexual reproduction. These are sperm-producing cells of a male crane fly, a species with only 8 chromosomes per cell. During meiosis, the cell divides twice, producing 4 haploid cells containing 1 copy of each chromosome. The first meiosis division separates pairs of homologous chromosomes, creating 2 haploid cells with only 1 set of chromosomes. The second division separates the chromatids in each chromosome. The end result is a production of 4 haploid cells, each with a single chromatid from each chromosome. When haploid sperm and egg unite during fertilization, the resulting cell is diploid, having received 1 chromosome of each pair from each parent.
Okay, I hope that was visible and audible to everybody, and I hope that that was interesting to watch. Okay, so with that, does anyone have any questions about meiosis? Because then we can move on to some questions that I've prepared. Just let me know. You can type your questions in the chat box also if you don't want to speak out loud. That's up to you. So is everyone good? Okay. All right, great. So then let's get started with some questions. So I'll read out the question and think about the answer, and you can answer out loud or send it in the chat. Up to you. Cool. So, first question. Why is meiosis necessary in sexually reproducing organisms? What do you guys think? Someone is saying 3. I guess you mean C. Hmm. It would be great if you guys could say it out loud also. But that's fine, actually. It's okay. Um, okay, so someone says C and then some other people say B. So, in fact, 2 people have said C and 2 people have said B. So there's a war. Does anyone have the final qualifying answer that can settle this dispute? Okay, we have the answer. So it is B. So it is to maintain the chromosome number across generations, right? As I mentioned, maintaining the ploidy is very important. When you have a fertilization event, you have N and N, and they come together, and the fertilized egg cell ultimately has 2N, right? And why it's not C is because, yes, of course, meiosis increases the number of genetic variations, but uh, not in initially sorry, not in the somatic cells, right? Initially, the genetic variations that meiosis contributes is to the gametes, so not in the somatic cells. Then gametes will combine later and form somatic cells, and those will have genetic variations, sure. But initially, meiosis is for the gametes. So that's why C is not entirely correct. It is B. Great. So, all right. Next question. This is very easy. If a diploid cell has two N chromosomes, how many chromosomes will each gamete have after meiosis?
N.
That's correct. And the people texting as well, that's absolutely correct. Everyone's got it right. It's N. It's option B. Great. So I don't think that really warrants too much explanation. So I'll move on to the next one. All right. This is a bit heavy. Which of the following correctly describes meiosis?
Option C.
Okay, that's one option. Anyone has any other answers? So a lot of different answers are coming in. Someone said C, then a person said B, then someone said A. So so I think I'll settle the thing. It's actually B. Uh, B is the correct answer. So why is that? Because DNA replication you have to remember the checkpoints per cycle. DNA replication will happen just the one time, and there are so many checkpoints that are important for that. So two rounds of DNA replication is out of the question. So definitely one round of DNA replication during S phase is occurring in meiosis also, in mitosis also. Now, what is special in meiosis is that there are two rounds of chromosome segregation, right? In meiosis 1, you separate the homologous chromosomes, and in meiosis 2, you separate the chromatids of a single chromosome pair. So, the option is B, the correct option. You have one round of DNA replication followed by two rounds of chromosome segregation. I hope that's clear for everybody, right? Okay, moving to the next one. In which phase? Okay. Oh, someone has raised their hand. Samiksha Kumari, do you have a question?
Sorry, ma'am.
Oh, okay. No problem, no problem. All right. Um, okay, fourth question. So, in which phase of meiosis does crossing over occur? Is everyone agreeing with the answers in the chat? So, there's people are saying it's C. Does everyone agree with that? Does anyone have any other answers? Hmm. So, A is not correct. It is C. The answer is C here because I mentioned, right, in the different phases of um, in the different sub phases of prophase 1, right? That's where the crossing over occurs. So the synaptonemal complex was loaded initially, and then it matured in pachytene, and that's when the crossing over event actually occurred. And pachytene is part of prophase 1, which is part of the first phase of meiosis. So the option here, the correct one, is C. Okay. So I hope that is clear now for everybody. If anyone has any questions, don't hesitate to ask. I'll move on to the next set then. Okay. So what is the significance of crossing over?
Ma'am, option C. It increases genetic variation.
Yep, that's correct. Good job. So everyone in the comments also, very good. That is correct. It increases the genetic variation. And yeah, none of the other options are even remotely correct. So great. Uh, okay, next question. So during meiosis 1, which structures separate? So think about this. Let me know the answer. Hmm. So people are saying one person says B and then two people say A. Does anyone have any other answers? So B is the correct answer here, not A. So maybe I need to go back and explain this a little bit more. So in meiosis 1, I mentioned, right? Let's just go back to how it looks, right? So in meiosis 1, I think this might make it clear. In meiosis 1, you have a homologous pair of chromosomes, and this homologous pair, so here this, let's look at this lower one, okay? So this is a chromosome, say chromosome 1, and this is chromosome 2. Now, in S phase, we double it. So we have a sister chromatid pair. This is chromosome 1 consisting of two identical sister chromatids. This is chromosome 2 consisting of two identical sister chromatids. What we need to do in meiosis 1 is to separate the maternal pair and the paternal pair of chromosomes. So these are homologous. That means uh, I'm so sorry. I shouldn't have said chromosome 1 and chromosome 2. These are homologous chromosomes of chromosome 1 itself. Okay, my bad. So let's start again. So this is 1 from the mom and this is from the dad. Okay, and then we just double them. And during meiosis 1, we want to separate these homologous chromosomes. So after some recombination has taken place during prophase 1, we want to separate this to one side of the cell and this to other side of the cell. So this entire homologous chromosome, this entire pair of sister chromatids, this entire chromosome goes to one side and this entire pair goes to the other side. So that means that the homologous chromosomes during meiosis 1, right, those are what are getting separated, not the sister chromatids. Okay. So sister chromatids will get separated in the upcoming phase in meiosis 2, which is very similar to mitosis. In mitosis, what would have happened? You have these chromosomes and they join, the microtubules will join here at the centromere and pull these sister chromatids apart, and then you have diploid cells and it's all great. But in meiosis, we have two rounds. So the first one will separate these two separate sides of the cell. Then you will separate the sister chromatids. Okay. So I hope that is clear now. Yeah. So during meiosis 1, the structure that separates the structures are the homologous chromosomes. So it's option B here, okay, not A. So I hope that is clear now to anyone who might have got it a little bit confused. Right. And why is it not the other ones? So sister chromatids of a homologous pair of chromosomes, that's not the case. That's what happens in a that's what happens in meiosis 2, right? Phase 2 of meiosis. And C is sister chromatids of non-homologous pair of chromosomes. That doesn't make sense because we're not separating the chromatids here at all in meiosis 1. And non-sister chromatids of a homologous pair of chromosomes, that doesn't make sense either. Again, one, because we're not separating the chromatids, and two, because we are separating homologous pairs, each pair separately. So the non-sister chromatids are going to be in separate sides of the cell. Okay, so I hope that was clear. It is a bit confusing, but I'm hoping that the visualization helped. Okay, so next question is meiosis 2. Why is it called equational division? Hmm. So people are saying A, but that is not correct, unfortunately. Yes, now someone says B. So B is the correct answer here, actually. So in meiosis 1, we have learned, right, in your coursework as well, that meiosis 1 is called reductional division because in meiosis 1 itself, the chromosome number is halved, right? You go from 2n to n. You're separating out the homologous chromosomes, the pairs. So essentially, you're going from, say, in a human, you're going from 46 to 23. What is happening in meiosis 2 is equational division because of B option, which is the chromosome number remains the same. So in meiosis 2, what is happening? You have 23 in, I'm just saying in the case of humans, okay? You have 23 chromosomes, not pairs, right? You have 23 chromosomes, and each has a replica of itself, right? Which is because you have two sister chromatids, two identical sister chromatids of each of the 23 chromosomes, right? And then these chromatids are separated from one another. So the number of chromosomes still remains the same because these two chromatids are exactly identical to one another, right? So that's why the chromosome number is staying the same, just the quantity of DNA is becoming half. That's why it is called equational division. So meiosis 2 is equational division and meiosis 1 is a reductional division. Okay. So I hope that is clear, crystal clear to everybody, right? If you have any questions, because this can get a bit confusing, please don't hesitate to put a question up. I can explain it again, or I can go back to the visualization of it. Yeah. All right. Okay, next question is how many daughter cells are formed at the end of meiosis? I've covered this one, so this shouldn't be too confusing. Yes, everyone's getting it completely right. It is 4. So option B is the correct one. We have 4 haploid daughter cells that form at the end of meiosis. Great. Uh, okay, next question. Which structure is responsible for holding homologous chromosomes together during synapsis? So I hope you remember what synapsis is. Yeah. Others also please participate. I'm seeing like the same 3, 4 names. It would be great if everyone could join in. Yeah. So anyway, the answer here is C. That's correct. It's the synaptonemal complex. Uh, so no, okay. So one person says A. So the reason it's not the centromere is I mentioned that when you have a chromosome that's duplicated in S phase, you will have two identical sister chromatids. And these two sister chromatids are joined to each other at the centromere. However, a whole different complex of different proteins that was taught in quite some detail, right? In, I think, budding yeast, in the synaptonemal complex, it was mentioned that there are these ECM11, HOP1, RED1, these different proteins that are part of this big synaptonemal complex that holds together these homologous chromosomes to one another during synapsis. So synapsis is when you have the joining, the aligning of these chromosomes and the crossing over event during prophase 1. So in those regions, that's where synapsis is occurring. That's done with the help of the synaptonemal complex. Okay. So this synaptonemal complex is a very sort of evolutionarily conserved and functionally conserved complex, right? And it's very important to load it at the right time and remove it at the right time so that the two homologs can be brought close to each other, allowed to recombine with one another, and then separated in time such that they can go and align correctly for metaphase and anaphase and so on. So it's very important for this this complex to assemble to hold together the homologous chromosomes. So this is not the same as the centromere holding together the sister chromatids. Okay, so I hope that's clear too. All right. Next question. Next question. What is a major difference between mitosis and meiosis? So that's great. To those who have joined a little later, people are putting the messages in the in call message chat box place, or you can say it out loud. Okay, and we're going through the questions like that. So please do participate. I'm hopeful that it helps you with your studies. Yeah. So the correct answer here, as everyone has already messaged nicely, it is option C. Meiosis produces haploid cells and mitosis produces diploid cells. Okay, so I think that is clear enough by now. So I'm not going to explain that anymore. Okay, and obviously the other options are deeply wrong because mitosis results in genetically diverse cells. That's not true. There's no crossing over really that occurs there. There can be some you know DNA repair that leads, if there was some DNA break, it can lead to some DNA repair and some slight changes, but that's within somatic cells, so it'll be within that tissue type within a couple of cells, so it may not necessarily lead to an entire new organism with genetic diversity. Yeah. So, anyhow, that's why that's wrong. And obviously, mitosis occurs only in reproductive cells, is not correct at all. Right. Just a second. Something is going on inside my room. Okay. All right. So, what is the name of the point where homologous chromosomes exchange genetic material? So, everyone is answering very nicely. Yeah, that's correct. So the answer is B. It is the chiasma. So I've sort of gone over this earlier. So great job for remembering that. It is the chiasma. That's where the recombination is occurring. That's where, as you read in your coursework, right, that there is this D loop formation, then uh, and these junctions can form where you have a template based sort of DNA repair process that allows this genetic diversity to be included. So yeah, that's correct. It's the chiasma. All right. Next question. Which phase of meiosis is responsible for reducing the chromosome number? So I think by now this would should be quite clear to everybody.
Yours is one.
That is correct. Very good. So some people, no, everyone is saying A only, I think. So yeah, that's correct. The answer is meiosis one. That is the reductional division phase of meiosis. Meiosis two is, as I discussed just a couple questions earlier. Meiosis two is equational division, where your genetic content is decreasing of each cell, but the chromosome number is remaining the same. Okay, so great, good. I think we've covered a lot of the important concepts here. So let's move on to a couple more questions. In which phase does the synaptonemal complex maturation take place? So I have outlined this earlier. I hope you guys remember. Anyone? Do you guys remember when the maturation takes place? People are saying A, C, A with a question mark. Hmm. So the answer, anyone else has any other answers before I go ahead? So the answer here is C. One person has said that. So it's Pachitine. Okay. Yeah, correct. It is C. So uh, essentially, loading of the synaptonemal complex takes place in zygotine. So I think that's why some people have said A. But maturation does not take place until Pachitine phase. Okay. So it's good to know that. So that's the answer here is C. So, all right. This next question. In which phase does synaptonemal complex disassembly take place? Good. So I can see that people are getting it right. People are saying B. And that is correct. So it is diplotin. So diplotin moving into diakinesis in both of these phases, you have disassembly of the synaptonemal complex. Okay. And it's very important that it happens on time such that the cells can align properly later on. So yeah. So loading in zygotine, maturation of the complex in pachytine, and disassembly in diplotin, and I think to some degree also in dikinesis. Okay. All right. Good. So next question. This is a true or false. Genetic variation during meiosis is brought about with the process of non-homologous end joining. Is this true or false? People are saying false. Does anyone think it's true? So yeah, that's completely right. The answer here is okay. So it's actually false. Non-homologous end joinings is actually inhibited during the crossing over and the double-stranded break repair. So it's actually homologous recombination that is encouraged during this process. Um, so not non-homologous end joining. Okay. So this one, the answer is actually false. Okay. Next question. The function of SPO11 in budding yeast meiosis process is. So what is the function of this protein? Do you guys remember from the coursework? I haven't gone into this while I was explaining, but I'm hoping maybe this is quite an important protein. I'm hoping you guys remember. Do you guys remember SPO11? I think maybe anyone? SPO11? No? So from your coursework, um, in the budding yeast meiosis process, and this is quite a conserved process, so I'm sure there are sort of homologs of this protein in humans and other animals as well. They'll just be called something different, but they function pretty much the same way. So SPO11 is a topoisomerase like protein, okay, and it's very evolutionarily conserved. And what it does is it actually is the starting point of this homologous recombination process. So what does it do? It induces double-stranded DNA breaks. Okay. So in a homologous pair of chromosomes, it induces a double-stranded DNA break. It can induce more than one double-stranded DNA break. So why is this double-stranded DNA break important? Do you guys know that? So essentially, if you want to initiate homologous recombination, right, you want to initiate this crossing over or this mixing of genetic material, you need to start at a point where there is some single-stranded DNA. Why? Because only then can you use the opposite chromosome as a template for repair. So unfortunately, I have not included an image here, but if you can remember the sort of homologous pair of chromosomes that I had shown in the earlier slide, maybe I can just go to that here, right? So if you have these homologous chromosomes, again, one from mom, one from dad, right? You have, say you have crossing over taking place over here. So what this SPO11 does is if the DNA is sufficiently exposed, and that can be with the help of different chromatin modulating factors that allow the DNA to be more exposed or less exposed to SPO11. SPO11 will create these double-stranded DNA breaks. And that creates this region, the small region of single-stranded DNA within the break. And what happens is that because the synaptonemal complex has caused these chromosomes, these homologous chromosomes, to come closer to each other. In fact, in your coursework, it was mentioned they come in and around 100 nanometers of one another. This process of homologous recombination using the DNA damage repair using the template of the same gene that's damaged here from the non-sister chromatid is actually enriched. It's actually encouraged. So if suppose in this region where I have put my mouse, if there is a double-stranded break, the template for repairing this will not come from here. Instead, it will come from here or here. Okay. So that is what that is sort of how the initiation takes place, and that's how it proceeds. So you have an initiation with these programmed double-stranded breaks, and then these breaks are processed. So you have some endonuclease activity that allows them to become single-stranded in those regions. Then you have DNA repair, right? And this DNA repair can occur with the homologous recombination process, um, where this D loop is formed, and then you can have a crossover or non-crossover pathway if you remember. And then eventually, after these intermediates are formed, this whole thing is resolved, and you have repaired it, but with recombination using a template from the non-sister chromatid. Okay, and all of this is started with the help of SPO11, which causes these double-stranded breaks. Yeah. So I hope that little sort of recap jogged your memory a little bit about what was there in the coursework, right? So this is quite an important protein, right? It's a topoisomerase-like protein. Okay. All right. Great. All right. I think I have a few more questions before the next section. So, which protein binds at the dsDNA break and forms a nucleoprotein filament? Again, this is all in budding yeast. Okay. You guys remember? Anyone remember what the answer to this question is? Like, can you remember the process? Maybe even if you don't remember the protein, do you remember why this nucleoprotein filament is important at all? You can totally answer on the mic, it's cool, or message it if you remember. Where does this come in? What is happening, in this process? I just explained it in briefly. So it's totally cool. I'll explain what it is. So the answer here is C, RAD51. Okay. So what happens initially here, right, as was quite nicely explained in the coursework, was that this SPO11 um, induces double-stranded breaks in the homologous pair. And then you have SAE2, right? SAE2 or I think there's one more, I think COM1 or something. That is what inhibits NHEJ. So earlier I said, right, that the non-homologous end joining process of double-stranded DNA break repair is actually inhibited. So you have proteins that come in to inhibit that. Then you have something that will essentially create from your double-stranded break some single-stranded regions where you can then um, which are basically recombinogenic, which are where this recombination takes place. So this MRE11, this is a 5' to 3' endonuclease activity uh, containing enzyme, and that cleaves the DNA from the 5' end, and that leaves this ssDNA overhang. So now we have these overhangs, okay, at the 3' ends that are recombinogenic. So what does RAD51 do? Which is RAD51 is also a protein. It binds over there at that ssDNA region and forms this nucleoprotein filament and helps with the search for homology. So what it does is it finds the nearby homologous chromosome and helps it invade into that. So I showed you, right, that there was a paternal chromosome, there's a DNA break, and it actually gets the template from the homologous chromosome, non-sister chromatid of the homologous chromosome, right? So this RAD51 binding to ssDNA, this nucleoprotein filament is what goes and invades into the non-sister chromatid region, finds a template there and forms the D-loop structure such that crossover can take place. So that's what RAD51 does. Okay. So I hope that is clear. Um, cool. So I hope everyone can still hear me. People stopped answering a while back. So, all right. Um, yeah. Next question. True or false? The template for DNA repair in one chromosome during meiosis is typically from the sister chromatid of the homologous chromosome. Now, I think you'll be able to answer this one. What do you guys think? Are you guys there? Hello? Anyone? No? Can you hear me? Just let me know. Okay, okay, you can hear me. All right, good. Okay, so people have said that this is true, and yes, that is very correct. This is true. So the template for DNA repair in one chromosome during meiosis, as I mentioned, is typically from, it's preferred, of course, from the sister chromatid of the homologous chromosome, right? So the other chromosome, the chromosome that is from, say, if the maternal, then from the paternal chromosome. Okay, and the bonus question here, if someone has an answer, is what is the, let's say, okay, someone has something to say? No? Okay, so what is it that allows this bias to take place? So, what is it that biases towards using the template from the sister chromatid of the homologous chromosome and not the identical sister chromatid of the same chromosome? What allows this bias to happen? I just mentioned it maybe a few minutes ago. So it has to do with sort of a distance and joining of stuff. So it is the synaptonemal complex. Okay. The synaptonymal complex is what helps form those regions of recombination and helps align the two homologous chromosomes together, right? And it brings them really close to one another, and that's how it biases towards using the template from the homologous chromosome and not the identical sister chromatid of the same chromosome. Okay, so the answer here to the bonus question is synaptonymal complex. Okay, right. Question 18. So I was talking about the D loop earlier. So in which stage does this D loop structure form, or where will you be able to see this D loop structure? Anyone? So what is this D loop structure? Right. Does anyone know what the D loop structure is important for? So it's important for crossing over, right? There was a crossover pathway, and then there was a non-crossover pathway, which has a double holiday junction forming in it. And that is where you have this template-based DNA repair and joining, ligation. Yeah, so you're right, Tanishka. It is C, prophase 1. Very good. So that is where recombination is taking place in pachytene, which is part of prophase 1. So of course you'll get to see this D loop in prophase 1. So that is sort of just a little extrapolation that you have to make there. So good, it's correct. It is option C. All right. I think this is the last question here. True or false? The holiday junction forms in the crossover pathway during recombination or DNA repair. The double holiday junction. Sorry, my bad. I should have written the double holiday junction. Do you guys think this is true or false?
It's true.
Hmm. So the double holiday junction actually forms in the non-crossover pathway. So it's actually not true. Hmm. So I think the way the strand invades actually is different with respect to the crossover pathway and non-crossover pathway, right? So in the crossover pathway, you have the ssDNA region that where the end is extended to a degree. Then you extrude the end. So the end comes out and you anneal to the extended end, and then DNA synthesis and ligation takes place. However, in the non-crossover pathway, where you see this double holiday junction intermediate forming, you have similarly it extends and all, but then you have two sides essentially on both the homologous chromosomes. You have this extension and then extrusion and then resolution of the structure. So actually the answer here is false. The double holiday junction forms in the non-crossover pathway during homologous recombination and DNA repair. Okay, so that was a lot of questions that we just covered. So let me know if you guys want a break or something. Otherwise, I'll continue with the material. There's a few topics that I thought I would cover, then a couple more questions, and then that'll be it. So does anyone need a break? Does anyone want to stop for a bit? You guys are good. Seems that everybody is fine. Vitality is good. All right, then let's move on. Okay, so uh, Hmm. Cool. So the separation of chromosomes, which we talked about quite a bit during meiosis, is and that happening correctly is very important, right? So in your meiosis one, you have your homologous chromosome separating, just to recap, and in meiosis two, you have the sister chromatids separating. Okay. And now in this case, you can see that which this is a very nice article if you guys want to check it out. Um, and it also encompasses what's seen in the coursework, which is that there are different ways in which microtubules from the spindle pores can attach to the chromosomes. Okay, and based on the attachment, you will have separation in a certain way. Now, based on the type of phase you're in, whether you're mitosis, you're in meiosis, meiosis 1, meiosis 2, the type of attachment that the cell would encourage is going to be different. Okay. So, in the case of mitosis 1 and meiosis 2, remember that these two are very similar in the case where you're separating the sister chromatids from one another. So this is a chromosome. These are the sister chromatids, one sister chromatid 1, sister chromatid 2. And what we want in this is to separate them equally. So one chromatid here and one chromatid here. So the type of attachment that we would want is is anyone? Bonus question. Does anyone remember the type of attachment that we would prefer? You can see it from this image only. It is amphitelic attachment. Okay. So amphitelic attachment essentially uh, okay, attaches the centrosome with the help of the microtubules to the spindle poles equally on both sides, such that the two chromatids, sister chromatids, are separated equally to two sides of the cell. Okay, and there are many of these spindle checkpoints and all that that make sure that that happens. So amphitelic attachment is what is needed for the sister chromatids to separate in mitosis and meiosis 2. All right, and there are some proteins that are important for inhibiting the other types of attachment that could occur potentially. So we don't want it such that this kinetochores, there's nothing attaching over here. We don't want that it is a merotelic attachment where one side these two are attached and the other side only one chromatid is attached. We don't want syntelic attachment where both the chromatids go off into one side. Right, so these are not what we want for mitosis and meiosis 2 because that would lead to some ploidy issues, right? That would lead to aneuploidy. So in this case, you have PSC1 which inhibits merotelic attachment, and you have this IPL1 that inhibits, make sure that syntelic attachment and this unattached kinetochore situation does not happen. Okay. And in the case of meiosis 1, now what do we want here? We want to make sure that the um, after all of this recombination and all that stuff has happened, we want to make sure that the homologous chromosomes separate. So in this case, what kind of attachment would we want of the microtubules to the with the help of the kinetochore complex to the centrosomes? Right? We would want syntelic attachment. Okay, so this is syntelic attachment. That's what we want. So we don't want the co-oriented homologous homolog joining. We don't want unattached kinetochores. This could all lead to ploidy issues, right? So you have other proteins that are important like monopolin, MAD2 and all that. They are important for inhibiting these other forms of attachment, and uh, and a checkpoint as well to make sure that there is proper syntelic attachment shown here of the homologs in a bi-oriented fashion, indicating that both of these chromatids will remain together. The sister chromatids will not be separated, and these chromosomes, after they've finished all their crossing over and all, will be separated. And there are checkpoints here as well to make sure that we do not have an onset of the next phase before it has properly been separated. So that is all about these types of attachments. It's another regulatory process on making and quality control process on mitosis, meiosis 2, meiosis 1. Okay. This is, I think these proteins are specific to budding yeast. There will be similar homologs in other organisms as well. Okay. So now, if we don't have proper disjunction, right? Just a sec. Huh. So if we don't have proper disjunction, then we can have a host of different ploidy related issues, right? So non-disjunction can occur during anaphase of mitosis, of course, which we have seen in earlier lectures, meiosis 1 or meiosis 2. During anaphase, the sister chromatids, or in the case of meiosis 1, the homologous chromosomes, typically will separate and move to opposite poles of the cell, and they're pulled by microtubules. So as you can see here, right, you have this in the meiosis process. This is normally what you have, and then you have all these gametes coming out haploid, evenly, all of them evenly haploid, right? In non-disjunction, this separation fails, and both sister chromatids or homologous chromosomes can be pulled into one pole of the cell. So what can happen? One set of cells will have too many chromosomes and one will have less chromosomes, right? So in mitotic non-disjunction, which I'm not going into too much detail here, you can have two aneuploid daughter cells. So essentially, one extra chromosome can just go into one cell. So you have both of the daughter cells. One will be 2n plus 1 and the other will be 2n minus 1, right? Now, in meiosis 1, if there is non-disjunction, this will occur when the tetrads, that is these basically the what are they called? The bivalents, right? You have the homologous chromosomes that are paired together. So those are or they're called tetrads or bivalents. They fail to separate during anaphase 1. So the end of meiosis 1, they'll actually, what will happen is there'll be two haploid daughter cells. One will be n plus 1 because it has an extra tetrad, and the other will be n minus 1 because it didn't get that tetrad, right? And both of these daughter cells will then go on ahead to divide once more in the next meiosis 2, and they'll produce four daughter cells, right? So two of them will be n plus 1 and two of them will be n minus 1. So I hope you understand, right? Because the ploidy will stay the same after meiosis 2. So if you have n plus 1 initially, you're going to have 2n plus 1 after meiosis 2. And similarly, the other set, when they'll undergo meiosis 2, you will have two of them, two daughter cells from here, which will both be n minus 1. So right at the start, this is the problem. Okay, so non-disjunction in meiosis 1, this is what will happen. Okay, so this is just for one homologous pair that they've shown. Remember that there are many other chromosomes. It's not like the cell doesn't have any DNA. This is just for the case of one thing. You will have both pairs going in to one cell, and then the daughter cell will not have. And then when you have meiosis 2, you will have the uh, aneuploidy in both, where this will be n plus 1 because you have one extra set here, and this will be n minus 1, right? Oh, but what if you have non-disjunction in meiosis 2? So that is the failure of the sister chromatids from separating from one another during anaphase 2. So since meiosis 1 proceeded fine, so you have two of the four daughter cells that will be completely normal, right? Because after meiosis 1, you will have two haploid, well, two whatever 23 chromosome containing cells, and then the other one set of them will be fine, and then the other set will be aneuploid. So this might make it clearer to visualize it. So again, you're starting here with the two homologous chromosomes. Each one of them comes into this cell because meiosis 1 has proceeded cleanly, and then you have a problem. Then it's like it pulls one chromatid extra into the cell. It does not divide. So these divided fine. So you had haploid cells with a single complement of DNA. And this one, because it did not divide evenly and both chromatids went into the daughter cell, one daughter cell, you will have daughter cells that contain this double amount of chromatids. So I hope that is clear. Okay. And of course, with these kind of issues, we can have all these different disorders that take place and then that show up as different phenotypes. So I thought I'd list a few of them that are well known, and you can read more about them. Of course, there are more disorders also that are available to read about. So then autosomal trisomy. So as you guys know, autosomes are essentially the somatic, uh, the non-sex chromosomes and the somatic chromosomes, and then you have your sex chromosomes, which are the X and Y. So in autosomal trisomy, you have this Patau syndrome, which is a trisomy of chromosome 13. So essentially, that means that the chromosome 13 missegregated, and some cells have an extra complement of it. Edwards syndrome has a trisomy of chromosome 18, and the most common viable aneuploidy, viable means that the person who has the syndrome actually survives well and is relatively healthy. Of course, there are some issues with, the mental faculties, and there are some health complications that come up, but overall the person with Down syndrome is healthy, normal, can survive, can just live day to day. They just have some learning problems. So this is the most common viable aneuploidy, Down syndrome, and that is the trisomy of chromosome 21. So this person will have an extra chromosome 21 in their cell. Okay, because essentially what would have happened is the egg would have had an extra chromosome 21, or the sperm has an extra chromosome 21, and when it fuses with the egg or sperm, correspondingly it will have there will be three copies instead of two, because there was some problematic segregation at some point. And there is some data to suggest that women who give birth at a later age, what is called, unfortunately, a geriatric pregnancy. So, like, the older a woman gets, the chances of these kind of segregation issues does seem to increase. Um, of course, now there are so many great genetic and testing techniques that can make sure that you know whether this problem is there or not, even before the baby has fully developed. So it's easy to tell if someone were to have a child at a later age, but it is something that can accumulate as a problem in an egg with time. So there, yeah, so that's Down syndrome. Then there are sex chromosome trisomies as well. Klinefelter syndrome, where you have an extra X chromosome in a male. So you have XXY. You have triple X syndrome, where a female will have a whole extra X chromosome. So you will have 47 chromosomes with one X extra. But what's interesting here is that because X chromosomes typically that are extra anyway are inactivated as bar bodies, instead of having, so in women we have, right, we have like a X chromosome, and then because we are double X, one of them is actually inactivated as a bar body. So here you will just have two bar bodies, and it seems that patients with triple X syndrome actually don't have much clinical abnormality. So it's not a lot of people get diagnosed. XYY syndrome is when you have an extra Y chromosome. And here also there's not a lot of clinical abnormalities. So a lot of cases just go undiagnosed. And finally, another example of a sex chromosome monosomy, and this is one of the only viable sex chromosome monosomies, or viable monosomies altogether, I think, which is Turner syndrome, where you have a monosomy of the X chromosome in a female. So you have just one X chromosome. So no bar bodies will be seen in this case. Okay. So those were the different types of cases of aneuploidy and polyploidy and all that. And I thought I'll talk about one interesting thing. There is something, because I work in the field of cancer, this is something that I have come up against a lot when I'm doing my reading. That there is a lot of aneuploidy in cancer. So aneuploidy, as we know, is an abnormal chromosome composition, and it's a major contributor to cancer development and progression. And apparently, according to a couple of publications that I looked at, 90% or so of solid tumors are basically a mass of cells that have divided uncontrollably and formed this solid mass somewhere in the body that is cancerous. These 90% of these tumors have some aneuploidy in the cells, in the tumor cells. And apparently, this aneuploidy is strongly correlated with a poor prognosis. What does that mean? That if there is a high degree of aneuploidy in the cells of the tumor, it can actually lead to worse outcomes for that patient who has the tumor. So that sucks. And then the selection for, so now how does this happen, right? How is it that you have aneuploidy in cancer? So we know that cancer cells undergo a lot of uncontrolled cell proliferation. They lose contact inhibition, they lose the ability to stop proliferating when it's no longer required. So there are a lot of mutations, like we had discussed earlier in a, I think, week 3 or something, that there's p53 mutations, right? So p53, the guardian of the genome, is very important for making sure that there aren't too many DNA, there's not too much DNA damage accumulating, and it can send cells that are too damaged off for apoptosis, or it can help with repairing cells. It can make cells like stop replicating until the DNA damage has been repaired. Okay. And if you have p53 mutations, you don't have anyone guarding the genome anymore, right? There are other proteins, of course, but p53 being a major one, when there is a mutation, there's a high chance that the cell can uncontrollably proliferate. So in that case, a lot of this handle on the sort of proper segregation of chromosomes also naturally also goes out of the window. So now you have tumor cells within a tumor, right? These cancer cells, as you can see here, that actually have varying degrees of aneuploidy. Some of them have extra chromosomes, some of them have less chromosomes. It's a mess. Okay. So they have shown here how these different chromosomes can be of different numbers. And how is it that the tumor, so this is just something that one of these articles here that I've shown has posited, that essentially if you have a lot of aneuploidy, why is it beneficial for the tumor, right? Is it something that the tumor would want? So it can help, or it can be um, counterproductive to the tumor, right? So this kind of based on a selection process, right? So based on a specific karyotype is basically the full set of chromosomes that someone has under the circumstance, so specific cellular circumstance. So whether there is some stress to the tumor cells, whether there is a hypoxic condition, whether there is some nutrient deprivation, whether it is in some tissue versus some other tissue, a whole bunch of different circumstances can help with selecting for the aneuploid cells that are actually sort of benefiting for the tumor, or they are negatively affecting the tumor. So you can have positive selection, say the cells which are aneuploid and suddenly they are proliferating even better because of that aneuploidy, they will be positively selected to make sure the tumor grows more and more and more. And then you will have negative selection, where say the cell, because of the aneuploidy, suddenly maybe the cell is becoming more apoptotic or is becoming more immunogenic and more visible to the immune system, then it's negatively selected. So it's like this tumor is this machine that's looking at the different aneuploid clones and figuring out, okay, this one is good and this one is bad. What is best as a net benefit for the tumor to keep growing, to keep proliferating, and to keep sort of taking over the tissue that is growing in. So it's kind of crazy. So I thought that would be something interesting for you guys to sort of think about, where, you know, aneuploidy in general is something that a normal tissue would immediately, you know, raise a red flag against and stop the cell from dividing. And if it can't be fixed, it would, you know, the cell would be killed essentially. It would go into apoptosis. In the cancer cells, it's so, the regulation is so changed, it's so dysregulated, it's so different from normal tissue, that aneuploidy is actually something that could be taken advantage of to improve the proliferation of the tumor. And the same thing is shown in the right side diagram as well, where you have a 2N population, and aneuploidy can arise, and then either it can be selected out, right? Negative selection based on if it's not needed, or it can just be maintained because it's not really making much difference. So you'll have some cells that are aneuploid, it's not really doing anything, cool. Then you can have mutations giving rise to aneuploidy. And then if there's a positive selection, that means that this is beneficial in some way or the other, and it it grows and outnumbers the non-aneuploid cells, so it spreads out, right? And then you can have uh, yeah, you can have a whole genome. Oh my God, yeah. So you can have a whole genome duplication as well, and this is possible, where you have an entire new complement of DNA in the cell, and in that you have aneuploidy, and then that spreads. So I thought it would be interesting for you guys to think about um, sort of the way in which cancers are utilizing aneuploidy, as well as many other sort of evasionary tactics to keep growing in the body, and how this is important so that we can understand the heterogeneity even within a tumor. Not because not every cell is going to be the same. They're going to have ploidy differences that will affect the way they divide. It will affect the cell surface markers on these cells, right? So that's another matter to consider when you're thinking about different therapeutic strategies to target solid tumors, that there is a lot of heterogeneity, not just in the case of the protein complements in those cells, but also in the genetic complement within the cell as well. Yeah. So, yeah, cool. So that's another point. Okay, so I have some questions here. So we can go through these questions. I hope everyone is still awake after that. Uh, Cool. So not too many questions left to go. Just I think two slides worth of them, and then I think one or two topics, and then we'll be done. Okay. So the preferred mode of attachment of chromosomes to microtubules during meiosis one is, do you guys know? I outlined this while I was explaining. I hope you guys are still there. Huh? Anyone? Question 21. Anyone knows the answer? No? So the preferred mode of attachment of chromosomes to microtubules during meiosis 1 is the syntelic mode. Okay. Yeah, just now someone is saying. Great, good. Yeah, it is A option, syntelic. Right? So where you essentially, as I had shown in the image, you have equal um, binding of the microtubules via the kinetochore to the homologous chromosomes, and such that the homologous chromosomes can separate. Okay, so that's syntelic binding, syntelic attachment. Yep. Okay, this one is a little bit of a reader, but which of the following are false statements? So read the statements carefully and let me know based on the options which one you guys think are the false ones. I've gone through, I think, all of these points, so I'm hoping you remember um, the stuff. What do you guys think? Hmm. So this is interesting. Almost every answer that is there has come in the chat. Someone says C, A, B, C. Anyone has anyone else wants to answer? So I'm glad you guys are giving it a shot. It's good to um, attempt these things. Okay. Uh, Anyone else wants to answer? I'll give you some more time. So, okay. So actually, the answer here is D. Okay, so everyone has answered everything except for D. But that's totally okay because I can understand that there might be some confusions. I'll just go through it with you guys. So the first one is ds-DNA breaks during homologous recombination occur at random. So this is actually a false statement. Why? Because it was, explained in the coursework that there are many processes that regulate meiosis. And to make sure that there are not too many ds-DNA breaks that can then not be correctly repaired, actually where the ds-DNA breaks occur is not random. There are some loci, some hot spots where it occurs, and particularly it's inhibited in the telomeric region and in the centromere proximal regions, right? So if you look at the question sentence number 3, frequent ds-DNA breaks occur in telomeric regions, that is actually false. It is inhibited in the telomeric regions because having a ds-DNA break in a region that has a lot of repeating sequences is actually not a great idea. Okay, it can lead to some problems when it comes to repair. So 1 and 3 are false. Okay, so the answer is D. And I'll just cover 2 and 4 as well, why they are true. So double standard break compensation allows redistribution of breaks. So it was taught in the process of regulating meiosis that these ds-DNA breaks uh, don't occur randomly, so they occur at sort of specific regions and they're not too close by. Also, they make sure with the help of ds-DNA break interference that these spots are not too close by to one another. Also, this DSB compensation, double strand break compensation, allows redistribution of breaks. That means there are some set hotspots where the ds-DNA breaks can take place more often. Now, what happens is if there isn't a ds-DNA break there for some reason, suppose there is some point A and point B and point C, and typically these are hotspots where ds-DNA breaks take place. Now, suddenly point A does not have one, does uh, does not have a ds-DNA break, and point B does not have a ds-DNA break. So what will happen, right? Instead of doing at C, they will find an A1, B1, C1 sort of a region that is uh, sort of maintaining that same distribution of breaks without being too far or too close to one another. So ds-DNA break compensation does allow redistribution of peaks. So the sentence number 2 is correct. And lastly, the 4, SPO11 is essential for ds-DNA break formation. This is in the case of budding yeast, and that is true. As I mentioned, it is very important for SPO11 to initiate this entire process of homologous recombination. And starting off, it is the topoisomerase like protein that causes these ds-DNA breaks. Okay, so 4 is also correct. That is why 1 and 3 are false, and the option correct here is D. Okay, but I'm glad that you guys tried it. I'm sure that you are learning these concepts now, अच्छे से. So, all right, next question. Easy one. True or false? During meiosis 1, it is preferable that sister chromatids bind to spindles of opposite poles. Is this true or false? What do you guys think? Hmm. So, yeah. So, actually, the answer here is false. Okay. So, during meiosis 1, it's preferable that the sister chromatids would bind to spindles of the same pole because we are separating homologous chromosomes, right, in meiosis 1. So, the sister chromatids, the identical sister chromatids of one chromosome, are bound at the centromere, and we don't want them to separate in meiosis 1. So the sister chromatids both bind to the spindles of the same pole, and the homologous chromosomes are the ones that bind to spindles of opposite poles. I hope that is clear for everyone. If anyone is confused, let me know. Okay. Okay, one more true and false question. Attachment of chromosomes is first cleaved at the centrosome during meiosis 1 and then next cleaved at the chiasma during meiosis 2. Is this true or false? What do you guys think? This is following from question 23. So if you think about what we discussed in that, as well as earlier on when we were discussing about recombination and the synapses and all that, you might be able to tell. Hmm. So I think maybe this one is a bit confusing, huh? Or you guys are tired, which I can understand. I am also. So I'll just go ahead with the answer here. The answer here is also false. Okay. So attachment of chromosomes is first cleaved at centrosome during meiosis 1. This is not true, right? Because during meiosis 1, the cleaving happens at the chiasma such that the so I think you remember um, from your coursework that there was an interesting thing that happens where the enzyme separase is involved and there's a REC8 uh, involved in uh, the cleavage of the attachments of the chromosomes by separase. And the question was right that how come the REC8 that is involved with holding these guys together, how come it doesn't get cleaved in both meiosis 1 at both the centromere as well as the chiasma, right? So there is some hypotheses as to how come that happens. There might be a spore protein involved, or there might be some interactions that help hold it together, or some phosphorylation events that protect REC8 from cleavage by separase. So essentially, what I'm trying to say is that uh, during the first during the first phase of meiosis, during meiosis 1, there is only one area that's cleaved, um, that is the attached region near the chiasma. Okay, that allows these two homologous chromosomes to separate. And in the next one, because we're separating sister chromatids of a single chromosome, it is cleaving at the centrosome, uh, just like in the case of mitosis, right? So this is false. Question number 24. This is false. It's actually the other way around. So attachment of chromosome is first cleaved at the chiasma during meiosis 1 and at the centrosome during meiosis 2. Okay. So I hope that is clear for everyone. Okay. Uh, Next question. So this is just a sort of whether you remember what I showed you guys. In Down syndrome, what is the karyotype that's expected in autosomes? Remember, karyotype is essentially the way to look at the full complement of chromosomes in somebody's cells. So in Down syndrome, what is the case? Do you guys remember which chromosome is abnormally segregated? It's good to know this one. So the answer actually is C. It's a trisomy of 21. So I hope this is useful. I got this from this website where it's showing the full complement of chromosomes, a karyotype of a person. This person, this is a female because they have two X chromosomes. This person has three uh, of the chromosome 21, right? So there was a missegregation, and ultimately the fertilized egg had three of the chromosome 21 instead of two. So that's a trisomy. That means there are three of them. So the answer here is C. Okay. Cool. So next question. True or false? This kind of goes back to what I talked about when it comes to aneuploidy and cancer. So is it true or false that aneuploidy is always a pro-tumor occurrence? So I was talking about selection, right? That how sometimes mutations or ploidy changes can be beneficial for a tumor to grow, and sometimes it may actually hinder because sometimes these mutations and all are quite random. So you don't have necessarily have a lot of control over whether a beneficial mutation, beneficial in this case for the tumor and not the person with the tumor, beneficial mutation for the tumor is occurring or whether it is not. So here this is also false because aneuploidy may not necessarily be a pro-tumor occurrence. If it is anti-tumor, if it is anti-proliferation, right, or pro-senescence or something, then the tumor might find a way to negatively select for that cell, and then eventually that will be outcompeted by other cells that are positively selected, that are performing better in the tumor. Okay. All right. Another one of the things relating to um, chromosomes and the disorders of segregation. So in Turner syndrome, what is the karyotype that you would expect in the autosomes? Oh, sorry. Why did I write in the autosomes? In Turner syndrome, what is the karyotype expected in the sex chromosomes? My bad. This should be sex chromosomes. Yeah. Anyone? So Turner syndrome is seen in women and seen in females where they have one less X chromosome. So it is a like people with Turner syndrome are healthy. They live quite fine lives. Um, they just have one less X chromosome. Okay. So the answer here is A. They have a single X chromosome. Okay. And this is an open question that will lead me to the next part, which is that homologous chromosomes pair up and recombination takes place during meiosis, right? But what about when you have an X and Y chromosome in males, right? X and Y chromosomes are not the same size, they're not the same shape. So how is it possible for them to recombine? How do they align? Where do they recombine? Right? They're not like the other autosomes, which are nicely, like look at one, two, three. These are all the same size, they have the same banding. How do the X and Y chromosomes, when it comes to homologous recombination in males, how does it happen? So does anyone want to try thinking about what the answer might be? Hmm. So it is a Interesting question that a lot of scientists decided to look into also. It is something that people have um, sort of studied and investigated in their work. And one of the ways this happens is recombination at pars. So pars are pseudo-autosomal regions. Oh yeah, someone said that. Really good. Shivani has got the answer. The pseudo-autosomal regions or pars are where this um, recombination between X and Y chromosomes take place, right? So they are morphologically and genetically distinct, right? The X and Y chromosomes. And during meiosis, crossover does need to take place, and you will have in the short arm, you have this par 1 in this region, and you have par 2. So par 1 actually is a lot more sort of a hot spot for recombination, and it's been seen that the loss of par1 has actually been observed to be associated with male sterility. And so that theory has come up that the existence of par1 is really essential for homologous XY chromosome pairing and the correct segregation of gametes. So, yeah, this par is where this occurs. So that's how X and Y chromosomes manage to recombine despite being very different in size and shape and all that from each other. This region specifically. Yeah. So that's about pars. I have a few more slides. I don't know. I think I can just quickly go through them, right? So this is just so this is all about the meiosis and all that. So now we're done with that. There was also a little bit of course content about nuclear organization, and I thought I might go into it in short. I hope you guys are still around. So as you know, the nucleus is organized very uh, meticulously with the nuclear pore complexes that allow import and export of various different things from small molecules that diffuse freely to larger proteins, RNAs, and all that require active transport. And there are these GTP, GDP based mechanisms for transporting various larger molecules across the nuclear membrane into the nucleus and out of the nucleus. And you have the lamina, the lamins, which are um, they are a meshwork of this duplexes of lamins that are found on the inner surface of the nuclear membrane, and they provide shape and support to the nucleus. And within the nucleus, you have these various different regions. You can have heterochromatin and euchromatin regions that code for different genes. Euchromatin genes very often are coding for very important functional genes, and they're typically a little more available or accessible to transcription factors. Then you have these subnuclear components, Cajal bodies, speckles, paraspeckles. You have the nucleolus, very important for RNA and ribosome synthesis. And these regions where, of course, you have the chromosomes that will condense during cell division. And the nuclear pore complex was explained in great detail to you, where you have these cytoplasmic filaments, and then you have the similar cytoplasmic filaments, but they are sort of encased in this basket-like structure here, a central pore. And all of this is encased within this nuclear envelope. So it's interacting with both the inner and outer lamina of the nuclear membrane here. So these nuclear pore complexes are very, very important and very, very complex multi-protein structures that help with import and export of different proteins and RNAs and molecules across the cell, across the nucleus. And so I had a few basic questions I thought maybe we could go through. I think there's like 5 or 10 questions here. So maybe we can just quickly jump through them. They're very easy. So what is the nuclear lamina primarily composed of? So do you guys remember what was taught? I think everyone is quite saturated by this point. So let's just jump through these a little quickly. Okay. So the answer here is lamin. These are lamin intermediate filaments. Oh yeah, correct. Very good. You're getting it. So the answer is lamin. All right. Which subunits make up the lamin filaments? Yeah, so the answer here is lamin A and lamin B. So this is just a sort of, so it's option A. Then this was mentioned. So what syndrome is caused by mutations in lamin proteins? Someone have, someone have the answer? So it's progeria syndrome. Okay. That's just something that was also, I think, covered in some earlier weeks coursework as well, progeria syndrome. And what is the primary function of the nuclear pore? We went through this just now. You guys can unmute and say also whatever is comfortable. So it is B. Yeah, correct. Good. So the answer is B. Transporting molecules between the nucleus and cytoplasm, both import and export, both passively and actively, right? All right, last few questions. Which of the following statements is true about nuclear pores? Yeah, so you're right. Very good. Everyone who's saying B, very good. The answer is that they require energy for transporting large molecules while smaller ones can move through passive diffusion. But that's not the only way, right? So that way they have a diversity in the way they can perform their functions. Okay, this is just a sort of I don't know, question relating to whether you remember something, which is the estimated molecular mass of the nuclear pore complex. It's a huge complex, so it's really big. Uh, so the answer really here is C, okay? It's 125 million daltons. Just imagine how gigantic that is. Like, it's so many proteins together. And in fact, people estimate it might be more also based on, if you include some more proteins that are there in the membrane and all. So think about how tough it is to sort of perform any experiments on proteins or try to do an immunoprecipitation of nuclear pore complex proteins when many of them can come down together. How will you resolve your gel? I don't know. Okay. So, what sequence allows proteins to be imported into the nucleus? So, there is some tag that you can add. Do you guys know? Right. So, the answer here is the nuclear localization signal. It's option B. Yeah, very good. Right. I think I'm getting these responses a little late myself. So, yeah, good. So it is B. It's an NLS. It can be present in anywhere in the gene, really. And we actually use NLSs also when you're doing, say, you want to target a protein artificially, then you can clone an NLS into your gene of interest such that, or your protein of interest that will then be, remain in the nucleus. And so that NLS would help it move to the nucleus. So That's a useful thing. And SRP actually is useful for targeting for secretion and stuff. Okay, last question. What role does RAN GTP play in nuclear transport? So I mentioned that there is active transport uh, that occurs, right, with the nuclear pore complex as well for certain molecules. So RAN GTP does something there. So as it's active transport, right, it provides energy for nuclear import and export. So it's option B, right? So based on whether RAN is bound to GTP or GDP. So GTP with the help of GEFs and GDP with the help of gaps, which are enriched in the cytoplasm or the versus the nucleus. You can have sort of a differential ability of RAN to bind to GTP, and that is what provides energy for nuclear import and export. Okay, so that is all of the questions. I hope that today was useful for you guys. Um, yeah, if anyone has any other questions, any other concerns, doubts, suggestions, anything you want, let me know. Otherwise, I think we're all good. Yeah. Okay. Thanks everyone for joining, and I'll see you next week. Oh, you're welcome.